Welcome back to Falcon Physician Review's online review for USMLE Step 1. This is Microbiology Module 26, Viral Replication. The overview here for this module is that each virus has to go through a certain series of steps in order to propagate and make progeny. They have to attach to the cell, get their machinery into the cell, get their DNA replicated into a way where it can make proteins, and then make progeny viral particles which can then be released. We're going to go through that process together, and that's what we're going to talk about in this module. Welcome back to Falcon Physician Review's online review for USMLE Step 1. This is Microbiology Module 26, Viral Replication. As you know, viruses cannot reproduce on their own. They require us. So what they do is they invade a cell, they take over the internal machinery to build their own enzymes and new viral structural proteins. They block the synthesis of host DNA, RNA, or proteins. And there's a sequence that viruses go through in their replication. It starts with binding, entry, uncoding, viral protein synthesis, replication of genome, and then assembly and release. And we'll go through those one by one. Binding. For a virus to attack us, it has to be able to attach to our cell surface. And viruses have specific receptors on their cell surfaces which target them to their host tissue. CD4 is found on T cells for HIV infection. ICAM is found in the upper respiratory epithelial cells for rhinoviruses. Parvovirus affects the erythrocyte P antigen, and influenza A binds to sialic acid. The presence or absence of receptors gives you your tissue tropism, which is why each virus attacks its own particular cell. Entry. Penetration differs between enveloped and non-enveloped viruses. If you're a naked or non-enveloped virus, you inject your genome or you enter via endosomes at the cell surface. If you're an enveloped virus, you enter via two methods. You can fuse your envelope with the membrane of the host cell, or you can enter via endosomes at the cell surface. Enveloped viruses fuse their envelope with the membrane host cell. So this figure shows penetration. You have the virus, like a DNA herpes virus or an RNA paramyxovirus. Its envelope receptors bind to the receptors on the host tissue, and then it fuses its membrane with the host membrane, leaving its own viral particles on the surface, whereas the capsid goes inside to do its job. Envelope viruses can also fuse with endosomes, as shown in this figure. There's an endosome at the very top opening up for the virus, and then the protons get pumped in. That causes the membrane of the viral particle to fuse with the host phagolysosome envelope, and then it releases the capsid into the DNA. You've got a DNA virus like hepatitis B, or your RNA envelope viruses as listed here. Uncoding is where the cellular proteolytic enzymes digest the capsid away from the nucleic acid. Remember, the nucleic acid is wrapped in the protective protein core, and so we have to have the proteins eaten away. This occurs in the cytoplasm. Replication of viral nucleic acid is a complex and variable process, and it depends on the nucleic acid type of this particular virus. We'll go through a couple of these. Protein synthesis and replication. RNA viruses undergo transcription, translation, and replication in the cytoplasm. One exception of this is orthomyxoviridae, which is segmented, and retroviridae, which changes its RNA to DNA, and then undergoes transcription and replication in the nucleus. There is an eclipse phase between the end of the encoding stage and the maturation of new viral particles, where no complete viral particles can be seen within the cell. DNA viruses undergo transcription and replication in the nucleus. The exception for this is pox virus, which replicates in the cytoplasm. Virus-specific mRNAs direct the synthesis of viral proteins. The RNA may be plus, minus, or reverse strand. Plus strand directly translates into proteins and enzymes, whereas the minus strand has to be transcribed using a viral RNA polymerase to change it from minus RNA to plus RNA, which then can be translated through the host machinery. Reversed strand is reverse transcribed to cDNA by the viral reverse transcriptase, then transcribed to mRNA using a host DNA-dependent RNA polymerase, and then subsequently translated into protein. Here's a figure which shows the different protein synthesis strategies depending on the viral type you have. In the top bar, if you have positive RNA, you can directly be translated through the host cellular machinery. If you have negative RNA, then you have to have an RNA-dependent RNA polymerase, which undergoes transcription to give you positive sense RNA, and that gets translated. If you have RNA, you require a viral reverse transcriptase to give you cDNA, and then you use the host to take that DNA to mRNA, which is then translated. Finally, if you have DNA, you have a DNA-dependent RNA polymerase, which the host has, to turn that DNA into mRNA, and then you get your proteins that way. DNA viruses are double-stranded with a plus and minus strand. DNA is transcribed into mRNA and translated into structural proteins and enzymes. The DNA viral mRNA is synthesized by the host polymerase. Single-stranded viral DNA, like parvovirus, is converted first to double-stranded DNA by the host cell polymerases. Let's talk about DNA virus replication, and we'll use the adenovirus as an example. First in the stage is adsorption, then you penetrate, the viral genome is uncoded, then you've got early transcription, which you use to make RNA. That RNA leads to early translation, which makes proteins. Then you have DNA synthesis and late transcription, late translation, which makes different proteins, which help for the condensation and maturation. Gene expression is divided into 2 or sometimes 3 phases. The early genes, which are transcribed and translated first, encode enzymes and regulatory proteins that you need to start viral replication processes. These are the proteins that the virus makes to shut off the host and turn all the host resources to making new viral particles. Late genes are different. They encode structural proteins that the virus needs for assembly of a mature virus, like the capsid proteins. Immediate early genes that are the ones that take over the cell and facilitate early gene transcription. They stimulate cell division. Differences between DNA and RNA virus replication are shown here, with the examples between the adenovirus and the poliovirus. So the big difference is after you get through the uncoding stage, you can both go to translation, and then you get synthesis of the viral polymerase, the RNA-dependent RNA polymerase, and you have inhibitors of cellular metabolism. The poliovirus requires the synthesis of an early viral polymerase to change its RNA into DNA. After that, you get a replicative intermediate, and then you can start doing your proteins. Whereas the DNA virus goes straight to translation and transcription, and they get their own synthesis. Early translation and RI or RNA viruses. Early translation of mRNA is brought about by an RNA-dependent RNA polymerase. This catalyzes the synthesis of a replicative intermediate, or RI virus, which is partially double-stranded, with one complete negative RNA strand and multiple partially complete positive strands. This replicative form shows up during viral RNA synthesis and has 2 complete strands, one positive and one negative. Some key things to remember for Module 26 include each virus has to have a strategy. If you think about it, and if you know what the viral genome is, whether it be RNA or DNA, and if you know the polarity, you then can make up a strategy by which you can get DNA to translate into messenger RNA to make proteins. And that's what each virus has to do. They have to adhere to their target tissue, they have to penetrate into the cell membrane, and they have to get the cell to make viral proteins for it so that it can make progeny and then bud and release. We're going to talk about the viral assembly and release in Module 27, which is coming up next.